Today I will be talking about glucose catabolism and the series of steps. So the first step is glycolysis. So this glucose is made out of 6 carbons, and in glycolysis this glucose is broken down into 2 parts, or in half, creating 2 pyruvates, which are made out of 3 carbons each. And so when this bond is broken, it releases energy. In this process, there's also ATP made, and then this molecule, NAD+, receives electrons, and so it will bond to the hydrogen, creating NADH, creating NADH. So this is the oxidized molecule, and this becomes the reduced molecule, and so the NADH will take the electrons and it will become an electron carrier. And so NADH will carry these electrons down the chain. So these 2 pyruvate molecules can undergo either fermentation or the fermentation pathway, or the respiration pathway. And so in fermentation, these 2 pyruvates the pyruvates will receive the electron from the NADH and will make acids and alcohols. And so when the NADH gives away its electron, it regenerates back to NAD+, and the pyruvates will take this electron and form acids and alcohols, and these acids or alcohols are made out of 2 or 3 carbon structures. So the organisms that undergo fermentation is because the respiration pathway is not available to them, or it doesn't exist, or it's not available, so they go with fermentation as the backup plan. And so these organisms undergo fermentation so glycolysis can continue, and they can continue making ATP to stay alive. So fermentation regenerates NAD+, so that glycolysis can continue. And in the respiration pathway, there's for the organisms that undergo the respiration pathway, there's a transition step in which a carbon is removed from the pyruvate, and so it forms an acetyl coenzyme and a carbon dioxide. And so these 2 pyruvate molecules undergo that take the respiration pathway and undergo the transition step, get broken down into 2 acetyl coenzyme molecules and 2 carbon dioxide molecules. And these 2 carbon dioxide molecules that are gases will float away and be released by the cell, and so we're just left with the 2 acetyl coenzyme molecules. and in this process here, during the transition step, the electrons that were in the bond here, that gets broken down, those electrons get passed down to NAD+. And so it's another redox reaction that occurs, forming NADH by receiving the electron from NADH+. And so these 2 acetyl coenzymes will then undergo the Krebs cycle, and the Krebs cycle is a series of reactions that break the bond in the acetyl coenzyme. And so the 2 molecules of the acetyl coenzyme, they will be broken all the way down to carbon dioxide, releasing that final bit of energy. And so the carbon dioxide molecules will then float away because they're just gases, and they will be released from the cell. And so in this process, NAD+ will be bonded to the hydrogen, forming NADH, which is an electron carrier, and then there's also another electron carrier called FAD, and this FAD will also receives electrons, and so it will bond to the hydrogen to form FADH2, which is, like I said, an electron carrier, and then there's also ATP being generated. And then which leads to the final step, which is a electron transport chain. And so in the electron transport chain, the NADH and the FADH2 deliver their electrons to the transport chain, to the transport chain. And so, let me see. Okay, and so this membrane here, as you can see, is embedded with proteins that are called the E-carrier transport proteins, and then also the ATP synthase. So the first step of the electron transport, electron transport chain, is the electrons being transferred from NADH and FADH to the first electron carrier transport protein. And so this protein will pass electrons to this protein, and this protein will pass electrons to this protein, and so forth. And so as each of these transport proteins receives the electrons, that comes with some form of energy, and that energy is used to push protons across the surface of the membrane. And the more protons or hydrogen ions that get pushed across the membrane, it starts to build a gradient of hydrogen atoms on one side of the membrane. And so this gradient is called the proton motive force. Here, as you can see. And so the protons will build up, and in order for them to reach equilibrium, they have to get back to the other side of the membrane by traveling through the ATP synthase. And so when protons travel through the protein ATP synthase, it's called chemoosmosis, which is basically the hydrogen ions moving back across the membrane. And it's driven by the proton motive force, which will also the proton motive force, and so that energy will that proton motive force that's pushing them across the membrane is a form of energy that it will cause the ATP synthase to spin, and so that that kinetic energy that has been formed from the ATP synthase protein spinning will be used to add a phosphate to ADP, and that will create ATP. So that's that. And then going back to the electron transport chain, the electron carriers deliver the electrons here, and they get passed down the line, and then they provide the energy for the hydrogens to be pumped across the membrane until it gets to the last electron carrier over here, which will be passed down to the electrons will be passed down to the final electron acceptor. And the final electron acceptor is a molecule that will receive the electrons. And if this is anaerobic respiration, then the oxygen is the final electron acceptor, and it receives the electrons and it becomes water. And if it's an anaerobic respiration, a non-oxygen molecule will be the final electron acceptor, and it will be the final electron acceptor. So for example, sulfate might receive the electrons and become sulfite, and the reason why it's It's called anaerobic respiration is because it doesn't use an oxygen molecule. And that is it. Thank you.